comes to molecular cloning, my method of choice is SLIC, which stands for Sequence and Ligation Independent Cloning. And unlike your traditional restriction enzyme-based cloning, which is kind of like cut and paste, SLIC is more like copy and staple. Basically, in restriction enzyme cloning, what you're doing is you're cutting two pieces of DNA. So you're like cutting an insert out of one place and cutting a vector backbone, and then you're stitching them together with DNA ligase. What happens with SLIC is instead of cutting the parts out, you're going to make lots of copies of the parts you want to join together using PCR. And when you do this PCR, you design your primers so that you're actually going to add a little bit of the overlapping region onto the thing you're amplifying. So our insert is going to have part of the vector on the ends. And now what's going to happen is we have at the ends of our insert part and our end of our vector part, we have regions that are the same. But these regions are kind of like hidden because it's double-stranded, so we chew back those ends a little, and now we have these kind of like sticky ends. And unlike the sticky ends in restriction enzymes, where we have those distinct, overlapping, matching parts that just need that one stitch to stitch them together, these overhangs are going to be kind of more like variable and loosey-goosey. But that's okay, because we're not going to try to stitch them together ourselves. We're going to make bacteria do it. So we take those pieces that we've kind of chewed back a little, and we get them to kind of stick in the right place, and then we put them in bacteria, and bacteria thinks like, oh, it's just damaged DNA, I better fix that. And so it tries to fix it, and it does fix it using its homologous recombination machinery. And so bacteria has all this complicated machinery, so why not just take advantage of it in the bacteria, which is where we're going to put these plasmids anyway. And so that's what we do. And so SLIC is really awesome, and you don't have to rely on there being restriction cut sites or anything like this. You can do it like with any sequence, all sorts of different things. So I want to tell you about it in detail, how it works, and go through the protocol step by step and things like this. So let's dive in. This is going to be a more technical post with more of the practical details. And so I'm not going to go into as much background. And so you should be familiar with molecular cloning before watching this video. If you're not, I have lots of content on that. But basically, when we're talking about cloning, what we're doing is we're taking a piece of genetic information. So we're taking like the instructions for making a protein, and we're sticking it into a workable form. And often we're talking about sticking a piece of DNA, so like an insert, into a plasmid vector. And so a plasmid is a circular piece of DNA that we can put in bacteria and get the bacteria to make lots of copies for us. And so the plasmid is like the circular piece of DNA. The vector is kind of like the backbone. And then you have your insert, which is the thing that you're trying to put into that vector to make this full plasmid. Although we often use the terms like vector and plasmid interchangeably, and it can get kind of confusing. But in the context of this cloning, I'm going to talk mostly about the vector being the backbone and the insert being the piece of DNA that you're trying to get in there. Now, there are multiple ways to do this. And one of the most common ways is going to be restriction cloning. This is kind of like the classical way. And I think that it helps you understand SLIC if you are able to basically understand what it's not. So it's not restriction cloning, but the principles are kind of going to be similar, but you'll see how SLIC does things differently. In restriction cloning, basically, it's kind of like cutting and pasting. You take typically what you're doing, I'm going to be talking about cloning in terms of like subcloning. So you're taking an insert out of one plasmid and sticking it into another plasmid. So you might be, say, taking it from some sort of plasmid that you just got from a repository, just a kind of generic plasmid, and sticking it into an expression plasmid so you can make protein from it. So what you need to do is you need to cut your plasmid that has your insert to cut the insert out. And then you need to cut the plasmid that has the vector plasmid in order to get your vector backbone. And then you want to join them together. When you're doing this cutting, you're doing it so that you're cutting them either with this exact same restriction enzymes or with restriction enzymes that generate complementary sticky ends. So basically, restriction enzymes are going to recognize specific sequences, and they're going to cut those sequences, and they're going to cut those sequences in a way that's going to leave an overhang. If those overhangs are complementary with one another, and you stick pieces of DNA with complementary overhangs together, then those can get stuck together. And then you can add a DNA ligase, which is like a joiner, and it's going to join those together, stitch them together, and then you can stick them into bacteria. And so restriction cloning could work really well if you have restriction sites where you want them. And so in some cases with plasmids, they often have like multiple cloning sites where they have a variety of different restriction sites that you can choose from, but you're limited by what's in the plasmid around your insert. Another limitation of restriction cloning is that you have to like gel purify out the pieces that you make, and you need to have a lot of the starting material. And so it's easy if you have those sites there and those sites aren't in your insert or anything. And it's a tried and true method, but I prefer SLIC, which is sequence and ligation independent cloning. Here we're going to do something similar. We're going to make two different pieces, and one of these pieces, and these pieces are going to have like overhanging parts that complement one another. But instead of generating these parts by cutting the plasmids, what we're going to be doing is we're going to just amplify the parts of the plasmid that we want using PCR. So in a test tube, we're going to make lots and lots of copies of the region that we want, rather than cutting that region out of a plasmid. And one of the benefits of PCR is that here you can basically add on the overhangs with your primers. And so you add on these overhangs with these primers that are going to match the vector that you want to stick it in. Now, when you amplify your vector, so instead of cutting the vector, we're amplifying it by PCR too. So basically, we just have to copy the part of the vector that we want, removing whatever insert was in there, if there was something in there. And now we have a linearized vector with these overhangs, which were already in the vector, but they're the overhangs of the primers from our insert amplification. So we were amplifying the insert with this extra bit on, the extra bit that we designed so that it would be complementary. It would be the exact same thing as this sequence. So this sequence we put on the end of our insert, going into the insert on both sides, one per primer. You have an overlap on each side, and that overlap is going to be the same sequence as you have in your vector plasmid. Okay, so now we have these two pieces with like identical ends. And now what we're going to do is, well, first we're going to add DPN1. I'll talk more about this later, but it's going to be degrading the template DNA. So we were using those plasmids as templates for amplifying or copying the region that we wanted. But we still have those templates in there. And if we leave them in there, they're going to mess with things, because what's going to happen is these templates, on the template, they're going to have the selection marker, so the antibacterial resistance gene. So they're going to be able to grow, and then you're not going to know whether it's your plasmid or the template plasmid. And so DPN1, it's going to selectively degrade the parent plasmid, because that parent plasmid is going to be methylated. And I'll go into this later. So now we've gotten rid of that parent plasmid, those parent plasmids, and we have our amplified pieces. They have identical sequences at their ends, but the identical parts are hidden in the double strand by the other strand. And so we need to chew back one of the strands to generate these overhangs that can then find one another and be like, oh, hey, we're the same thing. We complement one another. So we need to chew back those ends. Remember with restriction cloning, how we made those like precise cuts so they perfectly match? Well, with SLIC, we're not making precise cuts. Instead, we're kind of just we're just chewing the ends back. And not precisely, just enough so that they expose some single-stranded regions that complement one another. And we do this with actually using T4 polymerase, which sounds super duper confusing, because a polymerase is typically something that like it joins the letters together. So it's like you have a polymerase in your PCR mix that's actually joining together those deoxynucleotides to make that DNA sequence, those strands. But in the case of when you give this T4 polymerase, but you don't give it any of those deoxynucleotides, you don't give any of those letters, well, now it can't like put the letters together, it can't polymerize, but it can use its other function. And so its other ability is that it has an exonucleolytic activity, a 3' to 5'. And this is kind of like, it allows it to proofread as it goes. But if it doesn't have any letters, basically all it can do is chew back. And so it's an exonuclease. The restriction enzymes were endonucleases. They were cutting inside of a sequence. This is an exonuclease. It's going to kind of chew back the ends. If we let it go for a really long time, we'd just keep chewing and chewing and chewing, and we'd lose all of our sequences, and that wouldn't be good. So we just give it like 30 minutes. And then what we do is we give it one DNA letter. So we give it like DT, TP or DCTP or something like that. Doesn't matter, just one of the letters. And now when it hits that letter, it's going to actually stick that letter in there. But it can only stick one letter in, it can only stick that one letter you give it, and so it's going to get stuck. And it's going to, therefore, like stop chewing before it's degraded at all. So I'll tell you more about the design, but basically we design these primers typically so that they have like 20 nucleotides in their matching parts. And this way, as long as the T4 doesn't chew more than 20 nucleotides, we're good to go. But now you might be wondering, okay, well, those pieces are just going to be like randomly chewed, right? Well, what happens is we're not going to ligate them together in vitro. We're not going to add a DNA ligase, which was then just going to stitch together these breaks. Instead, we're going to stick it into bacteria just like it is. We're going to mix these pieces together and we're going to stick them into bacteria. And the bacteria, they have a lot more complex machinery. So they're going to see this as instead of basically, so they see this DNA that kind of looks similar to one another. And when things look similar to one another, then the bacteria go, oh, well, this looks like it's just DNA that got broken. And so we just need to fix it. And so because there are these matching regions, it can use homologous recombination. So basically relying on the sameness of them in order to say, okay, well, these parts are really the same thing. And so we just need this sequence one time, and we need to fill in based on the other strand. And so even though these ends are not going to be perfect, the bacteria are able to basically figure out what it's supposed to be and then fill in the gaps. And they can fill in these gaps, so they don't need to be like perfect. They don't need to be perfectly matching like you had if you were going to do it with the restriction cloning. Here you can have variable size gaps and things like this, but the bacteria can then use their homologous recombination machinery and do this. So I'm going to take you through more of the details of the protocol. I also want to mention that I've been talking about this in terms of subcloning. But there's actually a lot of different things you can do with SLIC, like mutagenesis reactions. So I talk more about this in another post, but basically with SLIC, you just need two pieces, and those two pieces need to have a matching part. And as long as you have those two pieces with matching parts, then what you can do is you can get the bacteria to stitch them together. And with those overlapping parts, kind of like placing those pieces together and figuring out that they're the same thing and creating your final product. And so SLIC mutagenesis is also really helpful. You can also design your primers so that they exclude part of the sequence. You can only like amplify part of the vector. Maybe you want to remove a tag or something like this. Basically, you can use all sorts of different strategies with your primers in order to stick any two pieces of DNA together as long as they have the overlapping region. Additionally, what you can do is instead of taking your insert from one plasmid, what you can do is you can actually take an insert from something else. And so it's getting more and more common for people to kind of like order gene blocks, they're sometimes called, which is basically you order some custom synthesized DNA, like a big chunk of DNA. So you order this big chunk of DNA, so it would be like just ordering this insert, and then you could just PCR it to make lots of copies of it. And if you had your PCR primers have the overlap, then you can make lots of copies of it that way as well. And in that case, you wouldn't need DPN1 on that part, but you would need it on your vector. Another thing, you can like cut your vector if you like still use restriction cloning to like cut your vector, things like that, if you want to linearize it differently. But typically what we're doing with SLIC is we're basically amplifying these two pieces. We're degrading the plasmid. We're then using T4 to chew back the ends, stopping T4 from going too far, mixing our pieces together, and sticking them in bacteria. And this will then allow us to do the subcloning. So in terms of protocol, I'm going to go into more depth about how I do SLIC. So the SLIC is based on basically, so the sequence ligation and independent cloning is based on this paper from 2012 from Mamie Lee and Stephen Elledge. It's really accessible in this like Methods of Molecular Biology book series. And this Gene blog page is really helpful. This article by Mary Gearing from 2015. It talks about kind of like the history of SLIC as well as its usage today. I should also mention before starting that SLIC is going to be often compared to Gibson, and it's similar to Gibson, but it's different. So in Gibson assembly and in SLIC, you both of these cases, you're kind of having this, the nice thing about these methods is that you can use whatever sequence you want. I mean, like you don't have to rely on there being a restriction site there. So that restriction site was fine if it was just subcloning, but if you were trying to do mutagenesis or something like this, basically it's not always ideal. And with these methods, you have this scarless cloning because you basically can use whatever sequence is there. You are using PCR to generate the parts you want to join together. Then you are chewing back those parts and joining them together. In SLIC, we rely on the bacteria to do a lot more. In Gibson, we do more of it in a test tube. And although we're doing more of it in a test tube, we're actually doing it in like fewer steps, but it's also going to be more expensive. So basically with SLIC, we are using the T4 polymerase's exonucleic activity. So basically we're just relying on its proofreading activity to generate those overhangs. And then we just, we want bacteria to do the rest for us after we basically anneal them. So we stick these pieces together so they can kind of figure out where things are going, but then they have these gaps, and then we put them in bacteria, and the bacteria fill those gaps for us. With Gibson, you actually do the chewing and the filling at the same time in vitro. And so basically you add this T5 exonuclease. So here it's not a polymerase, it's a dedicated exonuclease. It's going to chew back the ends, and you add and then you add a separate polymerase. So so this polymerase and this ligase, the polymerase is going to fill in the gaps, and then the ligase is going to stick them together, like piece them together. And so what's going to happen is it's going to basically do the fixing in the test tube before you stick it into the bacteria. But I'm going to tell you about SLIC. So Gibson is going to be more expensive. You have to buy their like these special products for it. So the SLIC is going to be cheaper. Gibson might be better if you are trying to do something with a complex sequence. So I know I talked about how the sequence could be like anything, but if you have a sequence that can form secondary structure or other problems, if it binds multiple places, I don't know. There are various reasons why you might have kind of problem sequences where you want the temperature to be higher. Gibson does a higher temperature. And so it might be better for that sort of thing. I also read that Gibson, though, is better for bigger inserts than smaller inserts. And so I recommend this J5 automated DNA assembly manual. They have some helpful information, and I'm not going to go into Gibson further, other than to say it's a similar thing, except you're kind of doing more of it in the test tube, less of it in the bacteria, and you have to use more expensive enzymes. So this is the SLIC cloning protocol that I use. This is basically based off of the protocol that I learned in Lemore Joshua Torr's lab during my PhD. And so let's just kind of dive in. So it starts with PCR. So we need to use PCR in order to make those copies of the region that we want. And we're going to have to design our PCR primers so that we have about 20 nucleotides of overhang. If you want to do more complicated SLIC, where you're like multi-pieces and things like this, they recommend in some cases using a longer overhang. And more of that in the original paper, they have some helpful advice for that. But typically about 15 to 20 is good. Aim towards 20 to be safe. You're going to want this overhang on your insert. I'm going to talk in terms of like insert and vector, but remember you can be doing like all sorts of different like pieces that you're putting together. You can actually do like multiple parts in the same test tube. There's some really complicated, cool
It's cool stuff you can use. But we're just going to talk about like the typical thing where you're amplifying your insert and you're amplifying your vector, and you're using primers that are going to generate overlaps. If you're just using a vector like that, you don't want to add anything to this vector other than your insert. Well, now basically you just need to make the insert have parts that match the vector as it already is. You don't need to add anything onto the ends of these the vector amplifying primers. So these can be generic for that vector. Now, this is going to be really helpful because you can then stick in whatever insert you want using the same primers to amplify that vector. So it's common if you're doing a lot of cloning, you'll have these primers that you can amplify the vector, and then you'll have specialized primers that you use to amplify the insert. But the vector-specific primers don't have to have any information about that insert, so you can use them with all sorts of different inserts, the same primers for that. So in the case of the insert, though, it doesn't have the overlapping region, and so we need to add it on. And the way that we're going to add it on is by adding it to our primers. So your primers for the SLIC are often going to be about 40 nucleotides long, with about 20 nucleotides matching the insert and about 20 matching the vector. I know I didn't draw this to scale, but basically you want to make it so that you have that part that matches the vector on the ends that are going in. I find the easiest way to design these primers is to use a tool like Benchling or SnapGene or CLC Main, some sort of like molecular cloning tool like software, where you can actually put in the plasmid that you want, make the plasmid that you want. So take the sequence of the plasmids that you have and just cut and paste it in the software. And then take the region, take your cursor and scroll over a region from like 20 nucleotides on either side of that insertion sequence. So that'll give you like a candidate primer. But that primer might have some issues. It might not be ideal. And so then what I do is I go and I stick it into, you can use like a primer checking tool or something like that. I use Amplifix. And basically you just can copy and paste your sequence in, and it'll give you like a quality score as well as the TM. And so what I do is I typically like watch this quality score, and if it's too long, what I'll do is I'll start trimming back the end of the sequence or making it longer. Make sure that you're trimming back the right end so that you're not like removing parts that you actually need. In the case of the primers that you use for your vector. In the case of these primers, it's less important. You can chew from either side, but just be careful that you end up with still about 15 to 20 nucleotides on either side. So these are going to be longer primers than you would use just for the vector. But anyway, so you go to that primer sequence, you copy and paste the region. You're going to need one per side, so you have one per strand. And now you can go and you can order those through a website like IDT or something. It's only going to cost a few dollars per primer. That's one of the great things about these is they're super duper cheap. And so now you've got your primers, you can go and you can run your PCR reaction. And so in our PCR reaction, our primer for our insert, part of it's going to match and then part of it's not going to match. But because each of these strands is going to start with the primer, you're going to get this extra bit added on to each of your copies. And so you end up with lots and lots of copies of what you want. Well, hopefully you end up with lots and lots of copies of what you want. You need to now like verify it. And so you can run an agarose gel electrophoresis, typically just like a 1% gel, run a few microliters of it on there. This is going to separate the DNA pieces by size. And so you should see that your bigger product, so like your vector, would be higher up. And then your smaller product, your insert, would be lower down. And then you can verify that the reaction went according to plan. If there were problems, if you see multiple bands, you might need to optimize your PCR. You might need to raise the annealing temperature to make things more specific, fiddle with the extension time, various things like this. I should mention too that this PCR reaction, you can kind of like use whatever your lab has. I would just use something with high fidelity. So you don't want it making a lot of typos because we want the sequence to be what we think it is. And so I typically use like fusion polymerase, but again, it's not, you can use different polymerases and just follow the standard protocol and that sort of thing. And so now hopefully you have pieces of the size you want and you can mix them together. It's important to know the sizes of the pieces that you're looking for, not just so that you can check based on the gel that they look good, but also because it's going to influence how much of the pieces that we mix together. So I typically keep like a construct log where basically for my SLIC reactions, what I can do is I can plan out which piece goes with which piece, which primers do I need to make the insert, to amplify the insert, which do I need to amplify the vector, and then which pieces go with which pieces to get this together. I did a ton of cloning in my grad school because I was working with this on this protein where I was doing a lot of mutagenesis because there were these 5 potential phosphorylation sites, and I was making all these different combinations of mutants to test various combinations of phosphorylation and things like this. So lots of PCR reactions, and keeping track of what pieces go with what things is really important, so that you don't accidentally mix the wrong things. So I also have on here the expected product size. So it's helpful to kind of plan things out like this. And you can see that some of these reactions needed some optimization, but some of them worked fairly well. And so then you just go and you optimize the PCR conditions. But hopefully all went well. And if all went well, well, now we're at this step where we need to add DPN1 to get rid of our template. So we have lots and lots of the parts we want, and we still have lots and lots of the parts we don't want. And so we want to get rid of those parts we don't want. Now, I should mention that if you do restriction cloning, basically you're not amplifying, so you need to have more starting material, because you're not going to be making copies of it. And when you cut, well, now you're going to get 2 different pieces. And so there you have to separate out the small pieces from the large pieces. And so this is done by you actually run all of your reaction on the gel, and then you cut the pieces out and you purify the piece that you want, and you've removed the piece that you don't want. With SLIC, we're not going to run a gel. We're only running a portion of our gel, a little bit, so that we can look at it and see if it's the right size. We're not running our gel to purify it. So this is not going to be a preparative gel. This is an analytical or diagnostic gel. And so as long as those products look okay, we're not going to do some sort of gel purification or anything like that. But we're not going to get rid of this way in the parts that we don't want. And so we don't have that 2 parts, but we have kind of the plasmid and we have the parent plasmid and then the part that we want. So with restriction, you'd have the 2 parts of the same plasmid. You kind of have it in 2 pieces. With the SLIC, you're going to have your template, and then you're going to have the template you were using, as well as copies of the region of it that you wanted. And so we want to degrade that template because, as I mentioned, that's going to have the antibiotic resistance gene. So if we were to stick it into cells, it would be that plasmid, it would be able to grow in the presence of that antibiotic that we're relying on to show us that the plasmid's in there. But we want to make sure that only the plasmid that we made is in there and not the one that we are using as a template. And so we're going to do a DPN1 digest. And I talk more about DPN1 in another post that I did the other day, but basically it's going to be able to recognize the difference between the DNA that the bacteria made, so all those copies of the plasmid, as well as the DNA that we made in the test tube. And this is because when bacteria copy DNA, they add this methyl mark, and this modification, and this doesn't get added inside. And when we do PCR and make copies in a test tube, you don't get this methylation. And DPN1 is a weird restriction enzyme. So restriction enzymes we talked about before were really, really specific, and they didn't recognize methylation. And so methylation can actually hide with their cut sites. But DPN1, it actually recognizes methylated sites. And so it's going to cut DNA up that's methylated, and it's going to really chew it up. And so this is going to degrade that parent plasmid And so then when we stick it into DNA, only the only plasmid in there, so the only thing with the antibiotic resistance gene, should hopefully be our plasmid. In terms of practical, so typically you want to do this at least like 2 and 4 hours. I like to do it overnight at 37 degrees C. So it's convenient to basically you have your reaction in a PCR tube from when you did PCR, and then you took a few microliters and ran your gel, but you still have all the rest of it in that pipette, in that PCR strip. Now what you can do is you can basically just stick that PCR strip in one of those, like in a dedicated, like a official rack, or just I like to make holders, makeshift holders out of by taping together the pipette tip racks, insert things, and then just put that in the plate incubator. If you're going to have to do it longer, like you can do it overnight, and then if you have to do it longer than just overnight, you can stick it in like the fridge. But otherwise you can go straight to the next step. And so yeah, so 1 microliter per 50 microliter PCR reaction. Okay, speaking of that next step, our next step is going to be PCR purification and DNA cleanup. And so what this is basically just using some sort of like spin column kit. There are lots of different ones. So this is like a Kia Quick PCR purification kit I was using in my grad school. And here we have like Zymo kits, like DNA cleaning concentrator and things like this. Basically just make sure that it's just some sort of like DNA purification column. And I have much more on how these columns work in other posts. But basically it just gets the DNA to stick and washes the other stuff off. In the case of our PCR reaction, we're going to have a bunch of salts. We're going to have DNTPs, various things like this. We're also going to have our primers, which some of these columns can actually kind of like selectively filter out. So basically those little primers are going to be more soluble under conditions that the longer pieces won't. And so you're kind of able to remove some of that smaller stuff. Okay, but that is going to be the PCR purification. And so now you've had this purified DNA. Typically what I do is I elute it with 20 microliters of water, and then I go and measure the concentration. So you can do this on like a nanodrop, nice and simple, or some sort of, that's like typically you're using a nanodrop, or there's like a qubit, there's just typical spectrophotometers, but nanodrops are pretty common these days. You should expect to see a concentration around like 50 to 100 nanograms per microliter. It's important to measure this concentration because later it's going to influence how we're going to mix these pieces together. But before we mix them together, we need to do that T4 reaction. We're going to chew back those ends. So you use T4 polymerase. And so basically we dilute the standard enzyme 1:6 in its buffer. So this is just the buffer. It used to come with like the buffer 2, and then you would add BSA separately, which is why I have like water added here. But in the case of the new buffer is like R2.1, which has a like some sort of recombinant protein in there, like BSA, or some recombinant protein in there. So you don't need to add BSA separately. But you add your diluted DNA polymerase, you add your DNA. So you're going to this is just going to be your entire PCR reaction. So it won't actually be 20 because you took a little bit of it for your gel, but it'll be close. And then you're going to basically add the T4. So give it 30 minutes at room temperature. You don't want to let it go too long or it's going to chew back too far. And then to stop it even further, you add 1 microliter of 10 millimolar DCTP or any other DNTP, that's the letter, so that it stops chewing but then it gets stuck because it can't go forward more than just that 1 letter. And then to even further inactivate it, you heat it up at 65 degrees Celsius for 10 minutes. So now you've got your T4 treated, purified T4 treated DNA. And so our next step is going to be mixing the pieces together and kind of getting those sticky ends to meet one another. And so remember, there's going to be like non-perfect gaps, but there's going to be the bacteria can deal with that. And so you still have these overlapping regions that can stick together during this anneal step. So for this SLIC, basically, this is called like a reaction, but really you're just mixing them together for 30 minutes at room temperature. So when you mix them together, you want about 150 to 300 nanograms worth of your vector. And then the amount of insert you have is going to depend on the size. Basically, we want to make sure that they're the same number of copies of the insert as there are of the vector, so like a 1-to-1 ratio. But our vector is going to be a lot longer than our insert. And so you might have a vector that piece that's going to be like 5 kb and an insert piece that's going to be like 1 kb. So you need 5 times as much of your vector as your insert in terms of the nanograms in order for there to be the same number of copies, because each copy of the vector is going to weigh more. So you want to do like a 1-to-1 ratio according to their length, which is their length is going to be their weight is going to be proportional to their length. And so we're measuring the nanograms per microliter, and we need to make sure that we have the like as many times bigger as it is, that's going to be how many times more nanograms we'll need in order to have the same number of copies. And this is where having those those tables that I use to like plan out my PCR reactions really helps. Okay, so now after 30 minutes, it's time for let the bacteria do the rest. So basically, remember, we're sticking this into bacteria, and the bacteria are going to think, oh, it's just damaged DNA. We can fix this. And they're going to fix it with their homologous recombination stuff. And so we need to get it into the bacteria, though. And so this is done through transformation. And often this is done with heat shock using chemically competent cells. So basically you take these weakened cells, you'll take out your tube of them, you'll mix your DNA in there, let it sit for a minute. And then you need to actually get it into the cells. So these cells are going to be like calcium chloride treated, and they're going to basically, they have their surface neutralized by this calcium, which is going to make it less repellent to the DNA. So although the inside of this membrane is going to be all lipidy, on the outside it's going to be negatively charged, and DNA is negatively charged. So they wouldn't get together, except that you have calcium in there that's going to help neutralize those negative charges so the DNA can get close. And once it's close, you're going to use heat shock. So basically you're going to open up pores in the membrane of the bacteria so that the DNA can get in. And so typically what you do is, well, the exact details are going to vary based on the cell type, but typically you're using some sort of like E. coli DH5 alpha cells. You do a heat shock where you stick it in the in a water bath, like a 42-degree water bath for 40 seconds, and then place it back on ice. Add 100 microliters to a milliliter of media. So I typically just use LB. If you're having issues, you can use SOC. It's a slightly, it's a richer media. And you're going to let them recover in the absence of antibiotics. So you stick them in your like thermal shaker for 30 minutes to an hour. So if you're using ampicillin, I typically just do a half an hour. If you're using kanamycin or chloramphenicol or streptomycin, with these, you want to give it like an hour. Basically, with these are translation inhibitors, whereas the ampicillin is not going to inhibit the translation. And so you're going to want to make sure that you allow these to recover and make the proteins they need to recover before you prevent them from making proteins. Whereas ampicillin is targeting the cell wall, so it wouldn't affect the ability for these cells to make those proteins they need to recover. Once those cells have recovered, you want to plate them. And so while they are recovering, you can actually get the plate out to pre-warm while the cells are recovering. And so that you're not putting them onto a cold surface once they've been warm for a while. They're all happy being warm, and then they're trying to recover, and you stick them in the cold. That would not be good. And so instead, basically, you want to plate the cells out. You want to plate the entire tube, but you have like a mill of stuff in there. And if you tried to plate a mill of stuff on one of these plates, it would just pour off. And so instead what you need to do is you need to pellet the cells. So spin them down in a centrifuge for like for a minute at like 10,000 RPM. Basically, you just want to pull those cells down so you can remove the old media. And now you can add on new media in a volume that you can then plate it all. We want to plate it all because our SLIC reaction isn't going to be entirely efficient, and the transformation isn't going to be entirely efficient. It's a lot easier for the cells to take in like supercoiled plasmids that have been copied in DNA, and they're all pure in bacteria, and they're all fixed and they don't need fixing, and the bacteria doesn't have to do anything, which is another reason why it's really, really important to do that DPN1. Our SLIC product, on the other hand, is going to be, we made it in this test tube. It's got all these gaps, and the bacteria are going to have to do work to host it. And so this is going to be less efficient in terms of how many colonies you would expect to get. And so you're going to want to plate all of it. And so you're going to spin this down, remove the old media, suspend it in less liquid, so like 100 to 150 microliters of LB, and then you can plate it. And when you plate it, you're going to make sure that you plate it on the plate with the right antibiotics. And so you want to make sure it has the antibiotics that are that your plasmid, the vector plasmid that had the antibiotic resistance for. So we don't care what antibiotic the one we cloned out of was, the insert. That insert's antibiotic doesn't matter. But the antibiotic of the vector, what's actually the product we're putting in, that's going to matter. We need to make sure that we're plating on the plate that has that antibiotic that the plasmid is going to be resistant to. And if you do this, basically you spread it out on your plate, you let it go, let it dry for like 10 to 15 minutes, or not like dry, but at least like let it sink in. Then flip it over and stick it in the plate incubator. So you want to stick those plates with the lid on the bottom so that any sort of condensation or things can drip down and not drip onto the plate. And now, so then you get those colonies, and now it's important that you actually verify those colonies are what you think they are. And so now you can do like colony PCR if you want to get a quick idea of whether it probably worked, but that's not going to show you whether the sequence was okay, but it can kind of weed things out. More on colony PCR in another post. But to really verify the sequence, you're going to need to send your clones for sequencing. More on this in another post as well. But basically, you'll get some results back. And you want to pay close attention to those regions where you wanted where the SLIC was actually happening, where they were actually coming together, because that's the place where you're more likely to get errors. But hopefully you don't get any errors. And so hopefully this whole process worked well for you, and you have the plasmid that you want. And so just to review, SLIC stands for Sequence and Ligation Independent Cloning, and it's a really awesome tool that we can use in order to stick pieces of DNA together as long as they have complementary sequences. And so we often use this for subcloning, where we're taking a piece of one plasmid out of that plasmid and sticking it into another plasmid, but instead of like cutting it out, we're making copies of these pieces. And when we make the copies, we add on a bit of the other thing so that they have overlapping regions. Once we generate these pieces with these overlapping parts, we then get rid of our templates with DPN1 digest. We then PCR purify out those pieces to get rid of all the salts and the primers and things like this. Then we use T4 polymerase. Although it's a polymerase, it has exonucleolytic activity. So it can chew back DNA. And so if you don't give it DNA letters, it's going to chew back. Then you give it one DNTP, it will chew forward, or it will not chew forward, but it'll basically add when it sees that letter, but it won't be able to go further. It won't be able to put in any other letters, so it'll get stuck. This is going to expose complementary overhangs. You then mix your pieces together. The pieces are kind of going to stick together, but there's going to be gaps. These aren't going to be precise little gaps, like no gaps. In the case of restriction cloning, you don't have any gaps, you just have nicks. Here you actually have gaps. And so the bacteria are going to need to fill in the gaps, and they're going to need to seal it up. But they've got the complicated machinery that they need to do this, the homologous recombination machinery. So when you take those pieces that you mix together and you stick them into bacteria, the bacteria can do the rest for you. And hopefully they do it all well, and then you send it to cloning to make sure. And so that is the basics of SLIC. And you can also use it for all sorts of cool stuff like mutagenesis. I use it all the time for that. But this is the basic idea. And so Hope that you found this helpful and hope that your SLIC reactions work well for you.
Video này do mình, Cương bên npslab.vn, tự chỉnh sửa phụ đề hoặc lòng tiếng bằng công cụ, thấy vậy thôi, chứ tốn công lắm á. Thấy hay thì nhớ bấm theo dõi kênh, cho mình thêm động lực làm mấy video phòng lab xịn sò nữa nha.